Welcome. Welcome, everyone. It's really a great pleasure to welcome you for this Emotions Brain Forum, for this edition in Geneva. We are delighted to be involved in this organization. 9 cities in the world welcomed events on emotions, with many researchers involved from many disciplines. And I'm very happy, in the name of the Swiss Center for Affective Sciences, to welcome all of you. So in the name of the center, I'm glad that you are all here and that we will have this event. I will just briefly say a few words about our center. So our center started in 2005, so already more than 15 years ago, with the ideas that if we bring together the disciplines that are interested in emotions, we can contribute to this new emerging field called affective sciences. So basically, affective sciences is the interdisciplinary domain that is interested in emotions and all the other affective phenomena. And to do this, we collaborate and we have researchers in the center who work in many disciplines, for instance, psychology, philosophy, neuroscience, also from the humanities, from history, also from sociology, from economics, affective computing too. The idea is really to share the same phenomenon, which is emotions and affective phenomena, and trying to understand it from many perspectives. And the kind of topics of research that are in affective sciences are, for instance, to better understand the elicitation of an emotion. So what are the mechanisms that allows us to feel fear, anger, also happiness, pride, boredom, interest, curiosity, etc. So the elicitation of emotions, but also what happens when we have emotions, what happens in our body, what happens in our brain, what happens in our behavior, and what are the consequences of emotions? So we know from decades of research that very often the effects of emotions are positive. They are very useful. They help us to pay attention to what is important, to better remember things. They help us to take decisions. So both at the individual level and at the collective level, we also look at the effects of emotions. So these are just examples of topic of research that we try to conduct with many disciplines involved. I just give you here the link in case you want to know more about our research in the center. And of course, if you want to contact us to have more information, for instance, on what we are doing. And in the context of our center, we were contacted by Viviana. And Viviana Kassam, who is at the origin of the idea of this Emotions Brain Forum. And later, Viviana will be able to give you some more information about the origin of the event and about the team who was involved in this adventure of creating these events. I would like to take also this opportunity to really acknowledge and thank our colleagues who were very strongly involved in the organization of this event. So in particular, Dr. Marcello Mortillaro here, who is the scientific coordinator of this event, Carole Varon, who took care of the communication for the center, and Marion Gumi, who really took very nicely care of all the administrative coordination and organization. And later on, I will also give the floor to Dr. Cristina Soriano, who is a researcher at the center, and also in charge of education and training here, and we will chair this afternoon all together.
Voilà, I would like to start by thanking all of you present in the room. I'm counting, eyeballing a good 150, 200 people. This is fantastic. I also would like to thank those of you watching us from anywhere else in the world, watching us online, and everyone welcome to this celebration of science, celebration of women, and above all, celebration of emotion, because that's what brings us all together. This is the 9th and last of a series of events all over the world that have brought together researchers, essentially in neuroscience, today is a little different, to discuss emotion research carried out by women. And there are good reasons why this is the case. And the president of BrainCircle Italia, which is the institution that's behind this event, will come in a few minutes to tell you all about the raison d'être of these events. Each one of them was devoted to a different topic. The topic today, bringing us together, is emotion, cognition, and behavior. And there are also good reasons for that, because emotions are tightly linked to the way we think, and that goes a long way into explaining how we behave. And emotions, contrary to what was believed for the longest time, can be the object of scientific inquiry in sophisticated and well-controlled ways. And they have been attracting the interest of more and more disciplines ever since the 1980s, and giving rise to what we now refer to as the affective turn, or the affectivism in general, even the affective revolution in scientific practice, which is finally bringing emotions back to where it should be, namely center stage in the study of cognition and of behavior among humans. There are many disciplines involved in this endeavor, and our director of the center, David, at the beginning already mentioned many of them. This event was mostly devoted to neurosciences, but in the spirit of our center, we wanted to complement that crucial viewpoint with other viewpoints from other disciplines. So we already saw sociology, we are also going to see psychology, and we are also going to see philosophy. Voila, and one more thing I need to say. Our menu is pretty full today. We have a lot of ground to cover, and we will not be taking questions from the audience after each individual talk. But we do have designated moments when we hope that we will have a chance to talk among ourselves and talk with our speakers, and that is during the coffee break, and then a little bit also after the conclusion of the talks. So I invite you to come and approach them, because they are not only knowledgeable, but very approachable. There is only one exception that I should announce already to this rule that I just announced. There is one of our speakers, for reasons totally beyond her control and totally unexpected, cannot be with us today, and that is Professor Julie Perron. But fear not, because she kindly agreed to have her talk video recorded, so we will still be able to see her, we will still be able to hear her, we will still learn about the cognitive and affective consequences of long COVID, as we had always wanted. Voila. And now, I don't want to take any more of the time of the important people today, who are our speakers. So we're going to move on to our next presenter. In this case, we have Lisa Bortolotti, who is professor of philosophy at the University of Birmingham. Oh my goodness, absolutely, thank you for, I'm missing an important detail. And this important detail, of course, is the president of BrainCircle Italia, Viviana Kazam, the origin of the idea. Please join me here and tell us about this wonderful initiative. Thank you.
Well, I am under the impression of a very strong emotion, and I'm going to say something that was not forecast before I tell you about our forum. And the emotion was listening to Eva Luz, and I agree with her ideas, but I must say that as a Jewish person and very linked to Israel, even though I do not appreciate at all Netanyahu politics, I feel in a way that taking again Israel as an example of evil hits me very strongly, and I don't know if other people that are linked to Israel and that are here felt the same. It is true, Netanyahu policy is a very nationalist, populistic, religious, and we all criticize it. But on the other hand, all over the world, there are much worse regimes than what is in Israel, which is still a democracy, thank God. And so I feel that my emotion now is to share this with you, even though I was not supposed to do so. Thank you. And now let me tell you why we are here today. This is the last appointment of our Emotions Brain Forum. And it started, I'm working in the brain field and in disseminating knowledge about the brain since maybe 15, 16 years. It started with the Hebrew University of Jerusalem, who arranged a meeting of brain scientists in 2006, I think. It was the first one. And I was shocked by the interest of this field that at that time nobody really was talking about. So with Rita Levi-Montalcini, whom I happen to know, we decided to make an association, BrainCircle, linked to the Hebrew University and to ELSEC, which is a center of neuroscience of the university, to disseminate this knowledge with a large public and also especially in young people, people not in the field, because we felt that brain regards each of us as an individual. After many years of organizing events, I realized that myself, I was inviting especially men. And I am very aware of women's situation all around the world. And I was wondering why. Why all the speakers in my events are mostly men, and also in most other scientific events all over the world. And I started to think, are there interesting women? Are there great women that could talk and are not given the possibility? And therefore, we started thinking of making a conference with women all over the world, the most brilliant and interesting scientists. And I was looking for a sort of fil rouge that would link all the, presentation. I didn't want to make the women conference. And we realized that emotions is a very important field of study for women. And this is very, very interesting because till about 30 years ago, emotions were still considered a bad quality of women's mind. This started with the Greeks and maybe even before. For Aristotle and Plato, women could not have a part in democracy and in society, could not vote, could not join society in a position of action because they were victims of their emotions. And therefore, they were not reliable. You couldn't account of them. They could have erratic behavior. And it took 2,000 years to get over this. And the study of emotions as a positive and important part of our behavior and cognition really started only about 30 years ago, together with the process of women liberation. Till we had a letter that we found out that I think was from 1968 in Italy, where a very important member of parliament said women could not become judges because they are so victim of their emotions that you cannot expect them to make a positive, objective judgment. How can they? And this was called grace against justice. Women should be beautiful, stay home, and not try to make justice. Obviously, in Italy now, over 50% of the judges are women. So this was very interesting. So we thought that we can sort of flip-flop, the stereotypes, and having women talk about emotions. And obviously, women have a great expertise of emotion. And I personally think that emotions are necessary now to face the challenges of the new millennium. Because for 2,000 years in history, history was made by men with a view of aggression and exploitation, which were the reasons why we became such a successful, in a way, part of the world. But now we cannot have aggression anymore, and so we cannot have exploitation of the world because the planet is dying. And we need a new expertise that is, in my opinion, an expertise of both men and women, which takes into consideration emotion and empathy as a very important role. So this is why we are doing this, and this is our last appointment for the time being. I must say that I enjoyed very much all the conferences. They are all loaded in our website, emotionsbrainforum.org. You can watch them. Women are fantastic speakers. They have a lot of empathy. They have a lot of connection with the audience. So it was a wonderful experience. And I think it brought also from the point of view of our counterpart, we organize with very important centers all over the world, mostly directed by my men. And at the beginning, when I said, let's have something with women, they said, can't we get some men into it? Why only women? And at the end, they said, you know, you're right. And we realize ourselves how it is important to bring women forward. And I would like to end this presentation of mine inviting you to watch a short, very short movie of Rita Levi-Montalcini, who was an inspirer, not only Nobel Prize and great scientist, but also promoting women. And this is a talk she had actually in the Hebrew University of Jerusalem with young women scientists, I think about 30 years ago. Enjoy it.
Nice to meet you. I come from China, and my question is following. What do you think about the importance of women in future research?
You know, if I understood your question, you have never to worry about this point, because it may change one day from you. Now women can do what I could not do when I was a young woman like you are now. So you have not to worry, or women should not, as the future may be very different from what we suspect it will be. So it is not important to believe, it is important always to act in a very ethical way, very rigorous, how you will behave, how you will face difficulty, how you will enjoy good moment, as it was for me.
When you started research, which problem did you face because you were a woman?
For women it was very difficult, almost impossible, to be a scientist or a social person and a good mother, good wife. Should they have no interest in being a professional worker, being women, they should be raised as future mother, wife and mother. I was against this age. So I say, I will never be neither a wife nor a mother. But I went to my way. So I did not decide I was not a scientist. I wanted to go with Albert Schweitzer to help against leprosy, which was very diffuse at the time in Africa. And I started medicine just to cure leprosy.
Rita, what qualities do you think a scientist should have?
I am not a scientist. I am more an artist than a scientist. My twin sister died, unfortunately, 8 years ago. Was a very twin, different from me, but excellent person and an artist. My brother was a great architect. I do believe that I confront life not as a scientist, but as an artist. Never forget that what's important in our life is to be perfectly correct, help as possible the other, never mind if you are a scientist or an artist. I do work a great deal for women in Africa, and they are very intelligent, but they have been always prevented from study by men who want to impose their own capacity, which is not better than women. Intelligence is equally well distributed between men and women, even if the approach of problem is different.
I'm from Italy, and despite my young age, I already had to go abroad working. But I have the dream to come back in Italy and help my country with my job. So you think one day I can come back and work in Italy?
Of course, you can do very well, because in Italy we have excellent people, top, really. We have no problem about intelligence. Italians are very intelligent. It's a gift from Italy and many other countries. So you can find, if you have any desire to know, you can come to see me in Rome if you want.
Do you have any regrets of what you've done in your life?
What?
About what you've done in your life, even though it's a really, you did really great things, but have you got still some regrets?
No.
Nothing?
Never.
Never?
I don't care about living long, doesn't make any difference. I do believe I had good luck to discover the injection. life does not end with your death. What it will survive to you is the way how you send program to other people. I mean, immortality is not your body, which of course is going to die too. I didn't care about dying, because what is important is the message that you give to other people. This is immortality of human being.
So when you discovered first NGF,
Yes.
were you thinking that it would have come to have such a big importance in all
No,
the possible
no.
fields?
I knew it was very important against the dogma of the time. I was against dogma, and I said, no, we are not programmed like an insect, because everything changed depending on the environment, if good or bad. The race do not exist, the racist, unfortunately, exist. The death of people, 6 million women of Jewish people die, because not they are of inferior race. Einstein certainly, nobody would think that is inferior race. He's a race.
This really was the best possible way to get us started. This message of enthusiasm, this message of freedom, of hope in positive change, of self-assertiveness and self-trust, and a message of inclusion is exactly what we believe in and is what we are trying to enact. And on top of that, she was right. Here we are. Mortality is about a construct as useful as the value of our message is, and her message is with us today, and we celebrate her, and she is still alive. And another way in which we are going to celebrate positive emotions is by the hand of our next speaker, Lisa Bortolotti. I will try not to mess up the slides. Lisa is professor of philosophy at the University of Birmingham, but she's also affiliated both with the philosophy department, obviously, but with the Institute of Mental Health in the Faculty of Psychology at the same university. And this combined expertise of philosophy and psychology also transpires in the fact that she's editor-in-chief at the moment of the journal Philosophical Psychology. Lisa is from Italy, like Rita. She studied in Bologna, in London, in Oxford, and finally in Canberra before arriving in Birmingham. Her main interests are in the philosophy of mind, philosophy of psychology, of psychiatry, biomedical ethics, and her main work has focused on belief, delusion, rationality, irrationality, and agency, a key construct that she's going to tell us all about. All of these are topics that have engaged her writing, and on them she has done 3 monographs and 2 textbooks. She's very prolific as well. From 2014 to 2019, she benefited from the prestigious ERC Consolidator Grant. This time was a project in which she investigated the potential beneficial effects of irrational beliefs, and then moved on to a very interesting project that we're going to be hearing about today, interdisciplinary, like we like to do, in which they try to provide advice for healthcare practitioners about how to empower, how to provide support and agency to young adults, adolescents, who were going through an episode of mental health crisis. To me, this is a fantastic example of how emotions are crucial, can be truly instrumental in empowering populations that are disempowered structurally because of an imbalance in power. So I don't want to take any more of your time, Lisa. I invite you to join us, and I invite you all to please help me welcome her. Thank you, Lisa.
Thank you very much for this wonderful introduction, and I'd like to thank BrainCircle for organizing this series of events, and of course the local organizers here in Geneva for having me. They've chosen to go with a philosopher. It was a brave choice. Let's see whether they've done something good. So The other thing that really moves me is to participate in an event that is dedicated to Rita Levi-Montalcini, which, of course, as a young Italian woman, was a very important figure in my life, and something that always provides inspiration. The project I'm going to discuss today is a truly interdisciplinary project. It involves two groups of researchers, a group of academic researchers, and each member comes from a different discipline, and a group of researchers which is composed of young people who have had experiences with mental health services. And we work together with them at all stages of the project, from the conception to the analysis of the data to the production of the results. And indeed, the main paper we have produced on this so far has a lot of co-authors. What we were interested in is interactions between young people who struggle with their mental health and seek support from emergency services, and the medical professionals who try to help them and support them. Why we were interested in this is because we knew that, there is a risk in all interactions that involve people with imbalances in power, that the person who needs the support, who needs the expertise, is going to be in a vulnerable position, especially if there are aspects of their identity that are associated with negative stereotypes. So in the case of young people who struggle with their mental health, we thought there was a layering of different stereotypes that might affect the way in which practitioners deal with young people. And indeed, before even starting analyzing the data that we had, we produced a paper where we tried to think about the risks that there may be in this kind of interaction. And the risks come from certain negative stereotypes that are associated with being young, but also negative stereotypes that are associated with having a mental health condition. We use a construct from philosophy that is epistemic injustice, which might sound like a mouthful, but actually it's a very intuitive concept. This is the idea that sometimes when we share our views, other people may dismiss them, not because they don't necessarily agree with what we are saying, but because they associated one aspect of our identity with a negative stereotype that makes us look less credible or authoritative. For instance, in my case, being a woman in some context could be such a stereotype, and indeed the philosopher Miranda Fricker, who developed this concept of epistemic injustice, she had sexism and racism in mind. She was thinking about those kind of situations that we've all experienced with, for instance, a woman politician in parliament stating something, as we had in the UK a few years ago, and the prime minister saying, "Oh, calm down." Reinforcing this idea that the woman was speaking because she was overly emotional, but her contribution was not something valuable. Now, the notion of epistemic injustice, I think, is extremely interesting because the thought is that we are all agents. So agency is just the capacity to intervene in our physical and social world and to pursue our interests and our goals. Of course, we don't always achieve our goals, but we have this potential to try and pursue the things that interest us. And we need support, of course, from other people, and we need everything else to cooperate. It's not just a question of capacity, but we do have this initial power. In the epistemic area, which just means when it comes to knowledge, we have the capacity to produce knowledge and to share knowledge, to contribute to collaborative projects. And in the case of young people, very often, there is an assumption of immaturity or lack of experience, they might affect how we take their perspectives, how much consideration we give to their contribution. And in the popular culture and in the popular press, sometimes we hear young people described as snowflakes when they show no resilience, for instance, or drama queens, the idea that they try to attract attention to themselves when that attention is not justified. And there could be, nothing further from the truth. I think the pandemic really demonstrated how amazingly resilient and creative young people have been, trying to find new ways of communicating with their peers in a situation that was absolutely unsettling for them. In addition, having mental health problems is seen as one reason why people might dismiss your perspective or might think that you don't have anything valuable to contribute, especially when, for instance, young people experience unusual beliefs or have distressing experiences, as it could be with psychosis or with depression. Their capacity to produce and share knowledge is questioned even further because the idea is that they don't have a good grasp on reality. So if they describe their experience in a certain way, we can doubt that description. However, that's not empirically justified. We know very well that even people who have experience of psychotic symptoms, who are depressed, are perfectly reliable and rational in most aspects of their lives. And the fact that they do have a mental health issue doesn't mean that they shouldn't be listened to or they shouldn't be given a voice, especially in issues that concern themselves and their own well-being. So our idea was to come up with a framework where we could help mental health practitioners to safeguard young people's agency, young people's capacity to contribute to knowledge and to share their experiences in the context of clinical encounter. We had an amazing set of data. It was videoed, recorded interactions, real-life interactions between healthcare professionals who were providing emergency services, mental health emergency services, and young people. And we observed those interactions with the group of academic researchers and researchers with lived experience. We looked at the transcripts. We discussed how the interaction was going and what effects that it had on young people. What we also did is using a method that is called conversation analysis, where we paid attention not only to what was said, but how it was said. So the gaze, for instance, whether the practitioner was looking at the young person directly, whether there were any pauses, any interruptions, what the bodily language was saying. And we noticed how the young people in the encounter reacted and responded to the healthcare professionals' attitudes. Whenever the healthcare professional was asking a question, for instance, wanting to know more about their experiences, they would open up and share a lot more about what was going on in their lives. But whenever the healthcare professional was trying to re-describe their experiences in a different way or was interrupting them or suggesting that they didn't really need support, the young person would close up. And unfortunately, these kind of reactions are go beyond the interaction itself. So we could also rely on interviews that had been done with the young people after the interaction, where we asked them what went well and what didn't go well. And we also noticed that when the need of the young person for support was not recognized and acknowledged, the young person mentioned that in a crisis they would know, they wouldn't go back to emergency services. So this has, I think, potentially really worrying health implications. So our main finding, I guess, it's not a surprise for anybody here, is that communication really matters. So how healthcare professionals talk to young people, whether they give them a voice, really makes a difference to how the interaction goes, and how the interaction goes makes a difference to whether the young person follows the advice of the healthcare professional and whether they seek help in the future. we came up with what we call the agential ladder. So it's just a ladder of 5 points that can help healthcare practitioners preserve agency in young people. You start from the bottom, and the first 2 or 3 steps are really the fundamental ones. Without those, you can't get to the top. The first is validation of experience. Validation does not mean that the practitioner needs to agree with everything that the young person says, but that they need to listen attentively to what the young person says and be curious about their experiences, because that's the only way that a healthcare professional can get the information that they need then to make a diagnosis and to propose some means of support. They also need to recognize that the young person has already been incredibly courageous in seeking help, and they need support if they get to that point. So dismissing their concerns, as unfortunately we've seen in many of these video-recorded interactions, is not productive. The third point is really interesting. Although young people appreciate when they're given a diagnostic label, because very often diagnosis is the key to further support and to find peer groups that can help them go through their situation, they noticed that premature labeling was very damaging. So without knowing exactly what the experience and the situation of the young person is, already stating that they are anxious or that they have depression may be very problematic. First of all, because young people have a very malleable identity. The young people we're talking about are in a phase of transition between childhood and adulthood. They may be changing schools. Their point of reference changes from the family to their peers. So they are in a situation where they're still finding themselves. Their sense of identity is still in transformation. And so being told that they have a condition that they may think of as something chronic or something that cannot be easily overcome will change their self-esteem and their view of themselves. So the idea is, don't think of me as a problem. I'm not just the problem. I'm not just a depressed person. I'm not just a person with schizophrenia. Think of me as a person who may benefit from certain types of support. The last 2 steps are the more, I would say, philosophically interesting, but also they couldn't get off the ground without the first steps. And this is really manifestation of agency. Initially, we were thinking about talking about responsibility in the young person, responsibility that would help them to take charge of their mental health journey. But we thought the responsibility was not the right concept, because our young researchers reminded us that very often responsibility is associated with blame. And very often in these interactions, unfortunately, we saw practitioners blaming young people for their mental health crisis or for the way they were dealing with them. So we thought that we needed a different concept, recognizing that we are never alone, we are never entirely responsible for what happens to us. And so we decided to go with affirming the capacity of the young person to contribute to positive change, recognize that maybe they've come already a long way by seeking help, and empowering them to think that there is more that they can do with adequate support, not by themselves. And finally, involvement in decision-making. The last bit of the interaction is usually about what comes next. What can I offer you? In that interaction, we have to avoid the practitioner just providing a solution already packaged for the young person to take home. We have to make sure that the interests and preferences and the history of the young person are taken into account in that decision. Of course, the young person doesn't want the practitioner to just give away responsibility for that part of the interaction, because expertise is fundamental, but they want to have a voice. So validation is about showing an interest in the young person's experiences and recognizing their need for support. And these are the top tips. They come directly from the people with lived experience who watch the videos with us. Just saying, I can see you're struggling, can make all the difference. I still want you to acknowledge my distress, even if you can't offer me services. Don't downplay or contest what I'm feeling. So this is super important. In some of these interactions, we really saw the practitioner trying to re-describe how the young person was feeling. There was one case of a young man saying, I feel miserable. And what the practitioner kept telling him is like, but you don't look miserable. You don't behave like someone who is miserable. Are you sure that's the right way of thinking about it? So they really attempt to make the young person see themselves differently. Just because I look okay doesn't mean I'm feeling okay. That really speaks to me because as many academics, I teach young people. And very often, some of these young people who were looking for support were being sent to emergency services by counseling services at university. These were university students, very articulate. But the more articulate they sounded when they were explaining their situation, the more adjusted, you know, the less the practitioner thought they would have anything to offer to them. But the fact that someone, you know, is articulate and can explain what's happening to them or has got family and friends doesn't mean that they don't need support from services. And finally, what we were saying earlier, communication matters. I can tell if you're taking me seriously by your body position and tone of voice, so everything helps. Very often we could hear patronizing tone, for instance, in the practitioner, and the young people pick on that immediately. So these are other quotes from our young researchers. You're really distressed, you are in a lot of pain. I think that kind of acknowledgement alone can be really powerful for someone who feels like they are completely alone, isolated, and they don't feel like they even have control over their own mind. Some of the young people that we interviewed said, you know, after a positive encounter, because there were also very positive encounters, this is the first time in my life I feel someone has really listened to me. So that was how powerful the encounter was for them. Avoid objectification is really trying not to treat the person as a problem to solve, which I think is a temptation, especially when services are under a lot of pressure with time and resources. So avoid reducing the young person to a diagnosis and recognize that they have complex needs. The way our young people were putting it is, don't put me in a box, you know. Acknowledge that I'm an individual and my story matters. Ask about myself, ask about my family, ask about school before labeling me. A well-founded diagnosis can be validating, but premature labels cause lasting damage. Explain that the label is just one part of the bigger picture. It doesn't exhaust the person's identity. And this is another quote from our young people. When you are a young person, your identity is so malleable, it's very easy for a label to become enmeshed with your sense of identity. You end up thinking there's nothing more to you than your mental health diagnosis. Life is hard, and instead of them dealing with it in a more holistic way, they've just been put into this box. It's like a simple explanation for something that has many complexities. Okay, so the last step was involving the person in the process of decision-making, which again doesn't mean that the responsibility for the decision needs to be on the young person who is already in a critical situation, but that the young person's perspective needs to be taken into account. So these were the recommendations from the young researchers. Take time to explain what solution you are recommending to me. Ask me if I've heard about the treatment option or whether I've experienced it before. Some of the young people in the videoed interactions had already a history of mental health services, so they knew that some types of treatments would not be helpful for them. I remember, for instance, one young person who was struggling with their mood after their mother died, and they were suggested grief counseling, and they said, I've tried that before. That's not for me. I don't want to talk about my mother over and over again. So this kind of contribution is really helpful for the practitioner to rule out some possible means of support that may not be suitable for some individuals. Ask about my concerns about treatment. It was another case where a person was not complying with medication. The person was actually blamed for doing that, but at no point the practitioner asked the young person why they weren't complying with medication, which also made it very difficult then for the practitioner to propose something else. Don't make up your mind about treatment before asking for my perspective. Don't ask me what type of treatment I need. So there is a very strong recognition that the healthcare practitioner is the expert. I prefer to explore and discuss this together. So it's a kind of collaborative enterprise, but it cannot be a collaboration if the point of view of the young person is dismissed. Be transparent about potential problems. I kept this for last because, as I mentioned, it was the most difficult for us to get around to. This sense that you can empower young people to contribute to positive change in their mental health journey without giving them entirely the responsibility for this. And the main point is really avoiding blame. It is problematic to blame people who struggle with their mental health for not complying with medication or for harming themselves. We saw a video that was incredibly distressing for us to watch, where a person who had attempted suicide was talking to a practitioner, and the practitioner kept making him feel guilty about the fact that he did that while in the house his sister was there, and his sister ultimately found him. And the practitioner kept saying, didn't you think about your sister's feeling, when you did what you did? And on top of, making someone who was already feeling very low, feeling bad about himself, what this did is communicate to the young person that his feelings were not important, that the feelings of everybody else were more important than his feelings. So it was very, very distressing to watch. So, it is important to refrain from blaming. At the same time, it is important to recognize that young people who struggle with their mental health can take some steps to improve their situation with adequate support. So, for instance, recognizing that they've already come a long way, recognizing that they've been brave to seek support is a first step towards that. So there can be more successful clinical encounters, and what we heard from young people researchers is that what they really need is empathy and curiosity. They need professionals to be interested in who they are and what their story is before recommending anything else or before labeling them in a certain way. So young people with distressing experiences and unusual beliefs can be treated like agents, like someone who can contribute to the conversation, who has a valuable voice. And they can, share their knowledge and make these decisions that the practitioner makes much better informed if practitioners remain curious and empathetic throughout the encounter. I think this is actually a bigger message than just youth mental health. I think in every relationship where there is a strong power imbalance because someone has the knowledge or someone has the access to services, it is important to think about empathy and curiosity as a way of safeguarding agency. Could be a mother and a child, a doctor and a patient, a student and a teacher, an expert and a layperson. It feels like the best approach is to let the person have a voice. That doesn't mean necessarily agreement, but it means that they feel valued and respected, and there can be more of a collaboration towards knowledge. I just want to acknowledge the team. So Rose McCabe, communication expert, was the PI in this project. Rachel Temple coordinated the young people researchers. Michael Larkin is a psychologist working on youth mental health at the University of Aston. Clara Bergen helped a lot with the transcription and the analysis of the transcription of the video recorded interactions, also background in sociology and psychology. And Matthew Broom is the director of the Institute for Mental Health at the University of Birmingham. He has clinical experience, he's also a philosopher. So as you can see, there is not just one academic discipline. It was really interdisciplinary group. And McPinn Foundation, they facilitate research by young people with experience of mental health services. They've been fundamental to us to be able to get this methodology off the ground. And if you want to know more about the project, we have a lot of open access resources, research papers, but it's not everybody's cup of tea, but also podcasts and other type of resources, public engagement resources that you can find in the project website. Thank you very much.
Thank you so very much for such a fascinating topic and research that you have carried out. And I think this is going to be useful not only to us who have access to adolescent, or it's not useful only for those who have mental health problems, but as you mentioned, indeed, this paradigm of empathy, of cooperation, of listening and respect towards others is exportable to any power imbalance communication, especially in the cases of interpersonal conflict, of course. So it was very valuable for all of us at all levels. Now we're going to do something a little bit different. We are not going to talk about emotion. We are going to experience emotion. Not that we were not experiencing emotion already. Viviana already told us to what extent we all are, but maybe a different set of them. And this is going to be thanks to the participation of an artist among us today. I'm referring to Olga Kokcharova. She was born in Siberia, but she now lives and works here in Switzerland. She's a musician, she's a composer, and more generally, she's a sound artist and also a landscape architect, by the way. She declares herself fascinated by anything that is audible. And so she works both with synthesized sounds, electronic sounds, and with recordings, field recordings, and their interaction as well. She has experience also working in multidisciplinary teams, and in this case around the topic of space and landscape design or landscape architecture, where she contributes her expertise as a landscape architect, but also as a music artist in sort of exploring the role of sound in creating spaces and in making us feel things as a result of the sound experience in those spaces. In her intervention today, she works in collaboration with a second artist. And here I'm referring to Annette Schmucki, who couldn't join us. She is a Swiss composer who was nominated for the Swiss Music Prize in 2015. And for her, the musical aspects of language are as important as the semantic content of the words. So she plays with the quality, the phonetic and prosodic features of language. And what she did, in this case for us, for this special occasion, was to take the abstracts that the different speakers had created for their talks, and she extracted the words and the phrases that called her the most, and then artistically recombine them in a form of contemporary poetry that she then created, 3 poems based on those abstracts. And then Olga, collaborating with her, recorded Annette's own voice with these poems and created something else. So we will have then Olga showing us the voice of Annette and what happens also when the voice leaves us. And we're all going to experience the sound and the silence within and outside the words.
Affective against an artist asking me you audition. Bass. Better between body and brain. Close communication, consolidation, critical, democracy. Describing, discussing, disgust, dreaming. Emotion fears to feel finally. Give me a glimpse. Hand in hand, humans illustrate and interpret. Leaders love less, my other overall. Perception, play, populism, proposal, quickly reaching the reliable role. Sense, significant smell. To talk, tender, to think and think through is underestimated. Vision, we, yet. encore une fois. Caught, please, dream, dream, body, feet, to fix, count, fate, love,
Welcome back. We are going to slowly but surely resume our celebration. And we are going to do so by moving in a different direction. We could say by going home to what the discipline was supposed to be in this type of phenomena, this type of symposia. And we're doing this transition hand in hand with Professor Hermona Sorek. She is professor of molecular neuroscience at the Hebrew University of Jerusalem and founding member of the Edmund and Lily Safra Center for Brain Sciences at the same university. She studied at the Hebrew University of Jerusalem, Tel Aviv University, the Weizmann Institute of Science in Israel, and the Rockefeller University in New York. Hermona's research pioneered the application of molecular biology and genomics to the study of cholinergic signaling. And this work has resulted in literally hundreds of papers, 60 of them at least published in high-impact journals. I'm really talking here, so that you understand, I'm talking Science, I'm talking Nature, I'm talking PNAS, I'm talking Neuron. Basically, there is nothing better. It is the very best of the best. She is elected president of the International Organization of Cholinergic Mechanisms and served as the elected dean of the Faculty of Science at the Hebrew University of Jerusalem between 2005 and 2008. And she has also been awarded honorary PhDs in Stockholm, Erlangen, and Ben Gurion University. In addition, she has received really prestigious prizes, including the Ilanit Katzir Prize for Outstanding Research Achievements in the Life Sciences, and very recently, the 2022 Israeli Prime Minister Emmett Prize in Neuroscience. Now, it is undoubted, but it cannot be doubted that Hermona is a world-renowned neuroscientist, but she's also one who dares to speak personally. I don't know if you've read the abstract or if you came here just because of her name, but she brought the notion of trauma, which is rather abstract and generic, close to us with personal experience, speaking to our hearts. And she is, I understand, passionate about what she does. So I'm sure that what she's going to tell us about, which is how emotional trauma can have detrimental effects on all of us because it can make us more vulnerable to neurodegenerative diseases down the line. This is a message that we're going to learn in detail and with passion from her. So please help me welcome Hermona Sorek.
Thank you, everyone. Thanks for coming back from the coffee break. I was a bit worried that what I talk about might be too technical for people who didn't learn molecular neuroscience. So I'll skip most of the technical details, and anyone is welcome to ask me later. So when we talk about the impact of traumatic experiences, I focus my research on one chemical, acetylcholine, which I'll tell you a little bit about, and the genes that control its function. And those genes are small, newly discovered, and very effective, and they function differently in men and women. I just realized that my talk speaks to the previous speakers in that, for example, the first emotion I thought about, just like Eva Illouz, was fear. But I don't fear exemption of particular human groups, which, of course, was quite exaggerated in that talk. Anyway, I would like to cite Seneca, who said that everyone is a slave of fear, and I think that that's what that lecture reflected to me. Acetylcholine was the very first chemical that was found to connect between nerve cells. Until Otto Lowy discovered it and won the Nobel Prize for that, people thought that the nervous system was a collection of wires that sent sparks of messages between them. And then it was found that there is a chemical. This is how it looks. And when the acetylcholine levels go down or up, we feel changed emotions. So this is what we are studying. We looked for the genes. We actually isolated the genes, encoding for the proteins that degrade acetylcholine. And for blockers of those, I don't know if anyone recognizes this picture, but this is Alexei Navalny, who was poisoned not too long ago by the same poisons that block cholinesterases. The problem is that other poisons are used as insecticides. So when we see an airplane spraying, you know what? If you see that, don't stay there. It's not good for you. Because we share with insects the same cholinergic network. And the picture you see on the board now is that of a brain of, I'm sorry, what did I do now? Okay, anyway. So that picture is that of a brain of people who live next to a spraying region and were exposed to insecticides blocking acetylcholine functioning. You see the red domain in their brain that says they are hyperactive, they are nervous. The blue region below says they have impairments in learning and memory. So as Eric Kandel, our friend at the center, says that psychiatry would never reach its goal until it considers the genes and their product. So, could changes in cholinergic genes alter emotions? We looked for that in individuals after corona infection. And yes, indeed, the cholinergic genes go down in the brain of people who suffered. One of the genes is called dancer, so I couldn't help myself looking for a dancer. Okay. But I'd like to talk about the impact of fear on people. So you can find papers on an individual who experienced an earthquake, developed a heart attack, and dropped dead. You can find a paper on someone who experienced a tsunami. And in most of these papers, the discussion says we cannot study that in an entire population because there are no populations that are consistently exposed to traumatic experiences. Well, I disagree. I happen to live in one. So we decided to look into the impact of trauma on people's health. And we decided to do that with the most ancient and simple method, just by checking one's pulse. And the Chinese knew that method thousands of years ago and developed it into a whole art. But it's so simple, it's so cheap, that medical experts don't use it that much these days. we decided to look at that in 18,000 individuals in Tel Aviv who are paid to come annually to a medical checkup by their employer. So they all work. There are Jews and Christians and Arabs, Mizrahi too, by the way, and they all come every year, and we ask them to volunteer filling a questionnaire about their trauma experiences. For example, like, to what extent do you feel the impact of terror on your daily life? Just mark 1 to 5. And we, in that case, are 3 students, or actually 2 students and 1 postdoc. Nadav Yayon is the one with the beard. He's now in Cambridge, UK. Shani Shenhar is in Tel Aviv University. And Nir Weisskopf opened a startup company. And when we exposed these 18,000 individuals, 4 years at a time, to her pulse measures, we got a number. And we used machine learning to find out differences in pulse. Now, a grown-up should have a pulse between 60 and 80. If it goes up consistently from 1 year to the next, and we had 4 successive years, that's an alarm, and it reflects an increase in the risk of stroke, heart attack, death of all causes. And we had people in that cohort who seemed that way. Their pulse went up, their cholinesterase activities in blood tests went up, and those were the ones who filled the questionnaire 5 out of 5 in fear of terror. So what we see now is that, being afraid is not good for you. I would like to send that message to Eva Iluz, actually. And the change was 8 to 12 beats, and we need to go back to the same cohort and find out the outcome. I would like to tell you about the story of how we sent that paper. We sent it to the Proceedings of the National Academy of Science, and Shani Shenhar was the first author, and she said, okay, submitted, what happens next? And I said, now every day you'll get to the website of the journal and there'll be no news. And one day you'll find, yes, the paper was reviewed and the reviewers want you to do this and this and that. You'll know what you'll do the next half a year. 2 months passed by and then there was a response, and it was 1 sentence: the authors should be applauded for performing this work. It only happened to me once in my life. So, yeah, and she's very, very happy with that. Okay, now let's go to an earlier age. If you take a young mouse, 7 days old, and you pull out their whiskers from one side, you get a depressed mouse. And these mice are humiliated by their members of the group. They stay outcasted forever, and we know the nerve cells in the brain that control that reaction, but we didn't know why that happens. So again, Nadav Yayon looked at that. The paper appeared last week in Embo Journal, so these are very new data. And what he did was to clear the brain of such mice 3 months later and develop a method to map their nerve cells, find out the processes they send, and compare that to the humiliation feelings of those mice. The model is very well known and established. And this is what came up. There is a collection of these cells. They all react to acetylcholine. They are all labeled red genetically. And we can, by this method, automatically identify the cells, count their processes, find out what happens. And what Nadav discovered is that cells change the way they send processes. They can either send 2 or many. What we discovered is that economically they have a limited amount of energy. So if they'll have 2 processes, they'll be very long. If they have many, they'll be short, because economically they can't do more than that. The important thing is we also developed a way to find out which genes they express. And those that express a certain gene called ELMO1, for God knows why, send many processes, and those that don't send 2 processes. So we now have a way to compare the structure of nerve cells, the neuronal network, and the genes it expresses. Okay, so if a mouse reacts that long, what happens to human beings with mental disease? And we did that by analyzing and comparing the reactions of men and women with either bipolar disease or schizophrenia, and look for the cholinergic system. I'm sorry, I'm addicted to the cholinergic system, and hoping to find an application. And this was done by Geula Hanin, who is a PhD student at the lab, today she's also in Cambridge, UK, and Sebastian Lobentanzer from Frankfurt, who I co-supervised, and today he's in Heidelberg. And here again, I talk with Viviana. So we, of course, remembered Rita Levi-Montalcini and her way of looking into cholinergic neurons. She discovered that nerve growth factor that makes them grow. But we could compare gene expression in a lot of patients between diseases and between men and women with those diseases. And what we find is that the response to those poisons that Navalny was poisoned with is altered. So the cholinergic system is in action again in mental disease. And we knew that the cholinergic system can block inflammation. So that was very interesting. The most important thing was that schizophrenics, men, shown here in blue, and women shown in pink, reacted totally differently. And bipolar patients reacted through the same pathways, but opposite direction. So we do have differences in patients with mental disease. I have a colleague in Tel Aviv University who keeps saying there is one brain, men and women. I'm sorry, no, there are differences. We are women and we have a different brain. So we wanted to find out the mechanism leading to that by looking at cell cultures. A lot of scientists use cell cultures, especially to look for genes and proteins. And we often look at those cells as test tubes. You never ask yourself if it came from a man or from a woman, except for exceptional cell lines. Like there's one cell line called HeLa, which memorizes Helen Lane, a woman who died with breast cancer, and her cells serve in research worldwide until today. And we remember her name because a book was written about her. But what I did in this study was to ask, are those regulator genes that we study the same in men and women, in cells, neuronal cells of men and women origin? And we found that many were male-specific, others were female-specific, similar or inverse. So there is a change in the cells. And by the way, do you know, 100 years ago, if a baby boy was born in England, they would dress him pink. And if it was a girl, they would dress her blue. And sometime it switched. Anyway, this is how our cells look like. And when we look at those genes that they differ, we find out that many which have little blue dots are reflecting schizophrenia. Other genes are reflecting bipolar disorder, and some reflect both diseases, but there are major differences. When this paper was published, I was asked to write a review about the difference between men and women in reacting to therapeutics. And that was written by an MD PhD student in the lab, Alon Simchovich, who's now specializing in cancer medicine. And what he found, which was quite amazing to me too, actually, I learned a lot from reading that text, is that we have differences in the concentration of drugs in the body and in the brain, differences in the side effects they cause. Drugs that increase bone fracture in women are a real problem. Differences in psychosis induced by drugs. And I'd like to mention to you one specific treatment in Israel, which is the same blockers of acetylcholinesterase that are used to treat Alzheimer's disease patients worldwide. And they are used in Israel for decades. And suddenly, 1 month, there was an announcement that women's dose is cut by half. Why did it take so long? Because women do not easily volunteer to clinical trials. So what happens is that we have drugs that were tested in male mice because it's cheaper. You don't need to account for the menstrual cycle. They were tested in human males, and they're put for treatment in the other half of humanity, which I believe needs to be changed. Okay. So here are the little genes we are focusing on. We know that there is DNA, which is transcribed to RNA, which produces proteins. Proteins do the job. And the genes encode proteins, but only 2% of our genome encodes proteins. The rest is the instruction manual. And that instruction manual encodes little genes that may control the function of the big ones. And those are, in many cases, primate-specific. For those who don't know, we are primates. So we developed a set of little genes that serve as traffic lights. They control gene expression, and they shaped our brain to be what it is. Okay. So what would happen if we stop the functioning of such a regulator gene? What we see is mice, and I'll cut the movies for you just to save time, that run very quickly all the time. You'd say, that's very good, they exercise. No, they run because they don't know what happens to them. And when we train mice to find apple juice at the other end of the maze, these mice turn around and go back, and they completely lose it. And they also suffer inflammation, which is also controlled by the same acetylcholine. Actually, by the Jewish tradition, feelings start in the gut. The Bible says, your guts are touched, not your brain. And the question was, does it happen in humans too? And Shanish and Har found that humans with inflammation express much more of that little controlling gene. But in the Alzheimer brain, we have very little of it. So we lose the control over other genes. And we can see that even before the disease starts, so that the cholinergic network is one of the first to be affected. And this is Mor Hanan, whose PhD was about this, and she just wrote me that she was promoted in the company she works in, so let's be proud of her. What about other microRNAs? I'd like to tell you a story, a Japanese story. In Japan, in 1997, the television broadcasted part of this very famous television program. And that night, 800 Japanese kids were hospitalized with acute epilepsy. So that raised, to me, 3 questions. First, what was wrong with these kids? Second, what protected the other 20 million Japanese kids who didn't get sick? And third, how come it happened so quickly? For a gene to be activated and get translated, it would take 48 hours. This was 7 hours. And I said, what if we had a controller gene that works in the brain and stopped working. And if that gene was one of those very small ones, it could explain the symptom. That was studied by Uriah Bekenstein, a PhD student in the lab, and Niba Mishra, a postdoc who came from India. And what they did was to engineer a mouse, that's what we do in the lab, where they hyperactivated the brain when the mouse had lower expression of that gene that we looked for. Then we looked in the Alzheimer's brain, and we found that Alzheimer's patients, 20% of whom suffer epilepsy, by the way, which is not really treated, had higher levels of that small microRNA, we call them, but it was not enough to stop their epilepsy. So here is one gene. In the case of ischemic stroke, the story is different. Here, what happens is that people get very sick, and they don't die from the stroke. Those of them who die because they develop pneumonia. Why? Because our immune system subdues activities to avoid penetration into the brain. So here, those families of microRNAs that we kept studying couldn't be helpful. So what happens when you need an emergency control of the process? What happens is that other RNAs get broken, and those RNAs, under normal circumstances, are responsible for translation. But now they are broken by proteins, and they create regulator sequences that we didn't even consider as existing. And we published that paper. This is Kasia Winek, who came from the Charité in Berlin for postdoc in Jerusalem, and now she's a young group leader in the Leibniz Center in Vienna. So what she found was that the family we studied before of microRNAs went down in the blood of stroke patients. And these new fragments came up, which we called changing of the guards. When you have an extreme situation, you need extreme means. And that is, of course, those that refer to the cholinergic system. Very last point. How early does it start? So this is Geneva Knin. She's a PhD student in the lab. And we are looking for the impact of stress conferred on pregnant mothers on the newborn babies. And we're looking at these new emergency small RNAs. And here is what happens in male newborns. This is when we study both of them. And now look at the females. There is a decline in these emergency controllers that affects only female embryos of mothers who got stressed during the pregnancy, which was totally surprising to me. And that's an emotion that we would like to continue studying. Shani is working on the manuscript right now, because most of these small controllers come from mitochondria, which we know we inherit from our mothers. So this is a real woman control process. Where do these small genes come from? They come from the brain through the cerebrospinal fluid, which goes down, the levels of which goes down with age. So that implies that we may be less protected against damages to the cholinergic system in aged individuals, which is what we are going to study next. And I went through a lot of material, and I need to thank first and foremost to all of the patient volunteers who donated data, field questionnaires, and some of them even donated their brain after death. Someone asked me once if we look at the brain of live patients. So the answer is now we collaborate with a neurosurgeon who got approval to take tiny samples of tissues from surgery sites. But everything I told you today was about post-mortem brains. We need to thank the foundations, and as Lisa said, the ERC award changed my life for 5 years. Unfortunately, it ended. And I would like to cover the impact of the cholinergic system as regulating emotions through small RNAs that control the expression of the brain in men and women differently, and we are proceeding with that process. I told you about small RNAs that are actually fragments of another small RNA that we knew exists, but we never guessed what it can do. I need to thank my family. These are my grandchildren, and they remind me that age matters, and that is important. And finally, I'd like to cite Marie Curie, who said that nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. And that would be my message to you. Thank you very much for listening.
I'm sure you agree with me. We were expecting passion and we got passion. And in addition, a whole cohort of data spanning all sorts of illnesses, all sorts of possibilities and gender differences, which is of course something that we need to be more aware of and more sensitive to. Thank you so much. Thank you for sharing with us in a very accessible way. Now we move on to a slightly different topic, although we still stay within neuroscience. Julie Perron is our next speaker. She is associate professor at the Faculty of Psychology and Educational Sciences here at the University of Geneva, where she holds the chair of clinical neuropsychology. She studied at the University of Rennes in France and then here in Geneva. And her career truly beautifully integrates both academic practice and clinical practice, if you wish. Her scientific interests concern the corticocentric bias in affective neuroscience. She studies the role of basal ganglia and the cerebellum in human emotions in pathological populations. But in addition, and hoping to anchor this scientific practice with her academic practice with clinical practice, she practiced clinical neuropsychology in university hospitals. First in Rennes, where she was first trained, and then here at the University Hospitals of Geneva, where she was promoted to head of the neuropsychology unit in 2019. So that means, importantly for us and for the content of her talk today, that during the pandemic, she was in charge of the adult neuropsychology unit at the Geneva University Hospital, and therefore leading a team of researchers that back then and still today work very closely with COVID patients. And what she's presenting to us today, albeit by video, but nonetheless powerfully, is the result, some of the results of that period of intense research on the effects of long COVID. So without any further ado, I give you by video, Julie Perron.
Hello everybody, thank you very much for the invitation. I am delighted to present the results of the COVID-COG project during this forum. As is often the case in neuropsychology, everything started with clinical observation. As of March 2020, we observed neurological disorders in the acute phase of the disease, and based on what we knew of neuroimmunological disease such as multiple sclerosis, we postulated the presence of long-term neuropsychological effects of COVID-19. In order to test this prediction made in March 2020, the COVID-COG project started on September the 1st, 2020, thanks to the Swiss National Science Foundation within the framework of the NRP 78. The COVID-COG project aims to study the long-term neuropsychological effects of COVID-19 and to provide health recommendations. 11 departments of the University Hospitals of Geneva are participating in the project, which is jointly led by the Faculty of Psychology and Medicine. More precisely, the aim of this project is threefold. First, to explore whether COVID-19 induces long-term cognitive impairments. Second, whether they are related to the severity of the respiratory form in the acute phase. And third, whether they can be explained by the affective effects induced by the pandemic context and or the lockdown measures. The COVID-COG project is a follow-up study. We planned to include 150 patients and see them initially 6 to 9 months post-infection, and then for a second visit, 12 to 8 months post-infection. We excluded patients with relevant medical history before the infection, pregnant women, and people above 80 years of age. They were divided into 3 groups according to the severity of the respiratory form in the acute phase, with mild, moderate, and severe patients. At each visit, they benefited from a validated assessment with neuropsychological, neurological, psychiatric, and olfactory assessments. They also benefited from an MRI with DTI and functional resting state sequences. We have screened more than 4,000 medical records, interviewed 300 patients, and finally included about 120 patients. To date, we have completed these inclusions at both visits. So today I will present the results of the first visit. The results of the second visit are currently being analyzed. So what are the main results of the COVID-COG project? In the first question, we observed the presence of cognitive disorders in a greater proportion than would be expected in the general population. These deficits concern executive and memory functions and an important self-reported fatigue. These impairments were associated with altered functional brain connectivity within the limbic system. So the so-called limbic system is known to underlie cognitive functions such as memory, emotion, and awareness. They have, as you can imagine, this disorder, this cognitive disorder, a negative impact on quality of life and functional capacity such as working activity. So secondly, we found no significant differences between groups, even if a greater proportion of patients in the moderate and severe groups displayed cognitive deficits. And thirdly, we found an association between certain cognitive function and secondary variables, for example, between memory impairment and olfaction, but no correlation with other psychiatric measures such as depression, anxiety, or post-traumatic stress disorder. So in addition to these main results, we observed some unexpected effects, once again thanks to clinical observation. So in order to illustrate these observations, I therefore suggest that we go through a clinical vignette. So Mr. L is a 51-year-old man, married, with one child, one adult child. He has completed a master's degree in engineering and currently employed as a programmer. He tested positive for SARS-CoV-2 8 months before the interview. He acutely manifested significant symptoms requiring intermediate care hospitalization for 10 days, so without intubation. So let's watch his interview.
Vous m'avez dit que vous étiez hospitalisé. J'ai besoin de savoir, voilà, quand vous êtes tombé malade, comment ça s'est passé pour vous et qu'est-ce qui a précipité votre hospitalisation?
C'était... Oui, j'ai eu quelques jours d'hospitalisation. C'était une expérience assez intéressante pour moi.
Ouais.
Ce qui était... Comment ça s'est passé? Vous voulez savoir exactement?
So in the acute phase, the patient remembers the hospitalization as an interesting experience. It appears that he struggles to remember how it happened and seems to lack attention. Let's see about his hospitalization.
Elle est arrivée dans le salon et elle m'a dit il faut partir, il faut aller à l'hôpital.
Et pourquoi?
Elle disait que j'étais bleu.
So the patient states that he was severely coughing, clearly lacking oxygen, and that his wife almost had to force him to go to the hospital.
Donc on est 6 à 9 mois après votre infection. Comment vous vous sentez?
Très bien, très bien. J'ai repris mon travail, je vous dis.
Ok, oui.
J'ai repris mes activités professionnelles.
Et puis vous remarquez pas de différence entre avant l'infection et aujourd'hui? Vous êtes le même?
Moi ça va, vraiment. Je suis...
Okay, so 6 to 9 months after the infection, the patient was feeling good and even went back to work.
La mémoire, la concentration, est-ce que vous, à ce niveau-là, vous remarquez des différences entre avant l'infection et après l'infection? Et aujourd'hui, en fait?
À quel niveau?
Au niveau de ce qu'on appelle les fonctions supérieures, donc de la mémoire, la concentration. Est-ce que vous remarquez des différences entre avant l'infection et aujourd'hui?
Non, moi ça va très bien. Je suis concentré.
So when being asked about his cognitive abilities, he confirmed that he felt concentrated and that everything was all right. Nevertheless, it appeared that he had difficulties to concentrate and remember things. So Mr. L has 15 to 35% of the patients included in COVID-COG subjectively reported few spontaneous complaints, but objectively had sometimes severe problems, particularly with memory. These symptoms is well known and very common in neurology and is called anosognosia. So anosognosia is a condition where you can't recognize other health conditions or problems that you have. Critically, we are suspecting that these variables best discriminated between the clinical profiles rather than the respiratory form in the acute phase. This lack of awareness regarding memory disorders was positively correlated to a lack of dyspnea, as well illustrated in the video except by Mr. L, but also a lack of awareness of olfactory disorders. We also investigated multimodal emotion recognition thanks to the Geneva emotion recognition task. We observed significant effects for irritation and disgust in the severe patients as compared to the mild. We demonstrated that the lower the recognition of emotion, the lower the olfactory recognition, and the lower the memory performances. No significant correlation with anxiety, depression, or PTSD was observed. So as mentioned earlier, this constellation of symptoms, which is called a syndrome in neuropsychology, is associated to brain functional connectivity modifications in the limbic system, so in the default mode network and the limbic system. You can see here the pattern of cognitivity. Ok, Okay, so what can we say about all of that? First, the impact of viral infections on the central nervous system, and more specifically in the limbic system, is something well known, actually. Limbic and associative brain structures, such as the hippocampus and the basal ganglia, contain more enzymes involved in inflammatory responses than other areas. On post-COVID syndrome, the involvement of the limbic system has been also demonstrated by other groups, by notably FDG PET study. Secondly, and regarding the nature of the neuropsychological syndrome, we are speculating that the common denominator would be an alteration of self-monitoring functions. These functions allow humans to permanently monitor their vital functions, such as respiratory or cardiac functions, but also sensory functions, olfaction, or cognitive abilities. This hypothesis would perfectly explain the phenomena of happy hypoxemia observed during the acute phase. So this would be explained, in other words, why people did not come to the emergency room quickly enough, as Mr. L very well explained in the interview. So finally, other phenotypes of patients exist, of course, and remain to be identified. We suspect the existence of syndromes that can be situated at each point of the continuum. On one of the extremities of this continuum, we can find profiles like that of Mr. L. And on the other extremity, we can find a profile that I will illustrate with a second clinical vignette.
Est-ce qu'au niveau de votre caractère, vous avez l'impression qu'il y a des choses qui ont changé depuis cette infection au Covid?
Oui.
Ouais, à quel niveau?
Oui, je me sens plus irritable en fait.
D'accord, ouais. Ouais.
D'accord. Patient.
Ouais. Au niveau du moral, ça va comment?
Ouais, justement, c'est pas. Je me suis même posé la question en fait, savoir si je devais pas un peu avoir un petit aide extérieure. Puis je pleure beaucoup, je pleure pour rien quoi. Enfin,
plus émotif que
ouais, j'étais pas comme ça. Moi, je suis avec notre métier, on en voit quand même en soins intensifs quand même. J'étais aguerrie par rapport à ça et puis.
Ok. Et pas d'épisode de désorientation. Vous savez plus du tout où vous êtes, qui vous êtes, quand on est. Que vous avez pu vivre après justement l'infection au Covid.
Oui, non, j'ai eu un petit épisode comme ça.
Alors à quel niveau est-ce que vous me racontez un petit peu?
Oui, j'étais quand j'habite là-bas, à plein palais, en rentrant, voilà, et j'avais plus le corps.
D'accord.
D'un coup, quelques jours, je sais plus où j'étais,
Vous saviez plus où vous étiez, vous avez l'impression d'être à côté de...
Ah, complètement.
Oui, d'accord. À côté de vous-même.
C'est bizarre. Ouais. J'ai des coups de fatigue.
D'accord, une fatigue importante.
Fatigue comme ça, tout à coup, qui me tombe dessus. Je dois m'arrêter dans mon travail et puis bon, je stresse parce que j'essaie de faire en sorte que je peux m'isoler, puis quand même, je ne remarque pas trop, mais enfin voilà, ça me pose un problème quand même. Comme en fait j'ai des petits problèmes de mémoire aussi, c'est pour ça aussi ce que je n'avais pas avant, hein, donc je perds, je sais plus, des moments je sais plus, donc je me dis maintenant il faut que j'écris tout. Chez moi il est possible partout. Alors. Mais justement j'ai oublié, dernièrement, j'ai oublié dont je vous parle, l'autre jour justement j'ai oublié mes courses dans le coffre, tout à coup, et puis aussi je perds le fil de la pensée, j'ai l'impression d'être un peu à côté de moi, enfin bon, je me retrouve pas, quoi. Je suis pas vraiment plus du tout, et puis c'est très bizarre de pas retrouver ces mots, de chercher toujours ces mots. Du coup, ça cause des problèmes avec mes... Enfin, j'ai l'impression, personne ne me le fait me remarquer, mais dans le cadre du professionnel, effectivement, c'est... je commence une communication, une conversation avec quelqu'un, avec une collègue, et puis tout à coup, voilà...
So, on the other extremity of this continuum, we find profiles of people with many complaints, a mild to moderate dysexecutive syndrome, hyperventilation syndrome, anxiety, depression, PTSD, and a major reduced quality of life. So we are currently working on these phenotypes. So this general hypothesis and the results of the COVID project, to our opinion, paves the way to a better integration of long-brain relation into the field of affective neuroscience. So I would like to thank my team, of course, my co-PI, Professor Hassal, and the Swiss National Science Foundation and the steering board of the NRP 78. Above all, I would like to thank the teams at the University Hospitals of Geneva who did not hesitate to actively participate in this research project while they were facing a dreadful pandemic. So thank you very much.
We really would like to thank Professor Perron for this effort to be here present with us today, even though she physically couldn't make it. And now we take a small break, just a few minutes, to submerge ourselves again into a different kind of experience, to try to recall maybe what we have learned in the past talks and to prepare ourselves for the final installment.
To accelerate, another apathy, to assemble, an artist asks to attack attention, a body as an assemblage of back, brain and bleed, a clear clinical code, daily damage deficits, to develop difficulties, discuss disease, disorders, dreams, to disorder, to dream emotions often embodied, event ever exaggerated, feel further, to grow, heard and to hear, known as loss, hide, matters of microbleeds, to lose neuronal network, parents, people, to play, to pluck reversed present, to regardless, really a lot, to rethink and sing, visiting white visitors, Norton, weird. Peut-être encore une fois de nouveau.
Sometimes words feel like rain. We're going to move on to a new sensorial modality. This time, not what comes through the ears, but what comes through us in other ways. In doing so, we will have the invaluable help of Professor Géraldine Copin. She is professor at Unidistance Suisse, an associate member of our Swiss Center for Affective Sciences here at the University of Geneva. She trained at the University of Geneva, where she did her PhD, Yale University, and the Max Planck Institute for Metabolism Research in Cologne in Germany. Her work looks at the psychology and neuroscience of odor and flavor perception. She has focused on olfaction during her PhD and then on gustation of food intake in the periods afterwards. But she's still interested in all chemical senses, perception in all chemical senses. Her work on these topics has resulted in more than 35 publications in high-tier journals, including Cell Metabolism. So in a nutshell, she studies the behavioral and neural correlates of food preferences and food choices, both in healthy individuals and in clinical populations. And here I'm referring obesity, type 1 diabetes, olfactory or gustatory disorders, etc. She uses psychological methods and also functional magnetic resonance imaging. And for all this work, she has been awarded the prestigious Excellenza Grant, which is the equivalent of the ERC here, given by the Swiss institutions. Geraldine's area of expertise is extremely topical today. In the wake of what we saw about long COVID, we know that another of the possible effects of COVID is indeed not being able to smell. We may wonder whether that matters or not, and she has the answer to that question. But this is just, of course, an anecdote. In general, what we want to now is learn more about the underestimated richness of the olfactory world. Thank you, Geraldine.
Good evening. Thank you for the invitation, and thank you, Christina, for the kind introductory words. So I'm going to try to convince you that olfaction does matter, and I'm sorry my nose is actually full. This is not on purpose, I swear. And I would like to be discussing with you, maybe start off with two questions that I would like to ask, and we can do this old-fashioned. You can just raise your hand. I would like to ask you first, if you had to lose one sense, imagine I'm telling you, you have to decide, you have to lose sense of vision, hearing, touch, smell, or taste, which one would you pick? So who here would rather lose vision? Okay, 3. Hearing? I see 3 as 4. Touch? Also a few? Okay, you have to pick one, remember? You cannot just go without one. Olfaction? Okay, majority. And taste? Also, okay, so you are totally compatible with what we typically found, which is about 95% of people would say, I would rather lose my sense of smell. Now, other question, who here would think his or her sense of smell is less accurate or less reliable than maybe vision or audition? Who thinks that? Okay, so not that many. And for those who believe this, I'm going to try to convince you that also olfaction is also really important for us as humans, not just for animals. And interestingly enough, this belief that humans are mostly visual animals comes from scientific ideas that are not based on scientific evidence. But for instance, Paul Broca, which is a famous anatomist, kind of brought up this idea that olfaction is not that important if we want to be rational, and actually that the brain would somehow have to leave some space for other function than olfaction. Or Freud is another person who really pushed for the idea that olfaction made us more like animals and less rational, but that's definitely not what we found in scientific studies, and that's what I want to work on today. And my talk is going to be divided in 4 parts. First, I'm going to give you a few scientific evidence on why our olfactory abilities are actually much, much better than we often believe. Then I'm going to move on to why olfaction is an important sense, especially in relation to emotions. which is probably why you're here today. Then I'm going to say a few words in this fascinating field, which is how we may communicate emotions or generally social information through body odors. And I'm also going to end this talk with a few words on what happens if you actually lose your sense of smell. So what do patients report when they lose their sense of smell? And not just COVID patients, but any type of patients, really. And if I can move on, olfactory and olfaction in general has critical roles for humans, and olfactory dysfunction do come with significant life impairments, which is what I'm going to describe to you. In terms of what are we able to do in terms of smell? There are more and more studies working on this, and we are able to detect a very large range of odors. It's hard to estimate exactly how many. It depends obviously on the assumption you're going to make. Generally, we think it's around 10,000 odors that we could be able to just detect. But overall, we would be able to detect any chemicals which is made of 1 or 2 atoms. So really, a lot of different odors. In terms of discrimination, they could go really up to 3 trillion different odors that we could discriminate. So this is really a lot. And obviously, we might not be able to detect the same odors as others, for other species, I mean. For instance, you can imagine that dogs might be much better than us at detecting urines on fire hydrants than us, but we might be much better than them at detecting different odor of wines, for instance. So this is not systematically studied enough, I find, but the few studies that did this really found differences, and depending on the compound, we might be much better than dogs, and dogs might be better than us, but it's not always as clear as you may think. And in terms of size of olfactory bulb, this is another thing that is often brought up, right? Can you see my mouse? Yes, okay. This is the olfactory bulb in the mouse and human, and you could see that in terms of absolute size, the olfactory bulb is actually much bigger than the mouse one. Of course, if you look at the relatively size of the olfactory bulb compared to the rest of the brain, it's relatively small. It's about 0.01% of the brain in humans. In comparison, it's 2% in the mouse. But if you look at the absolute size, you can see it's actually quite large. And in terms of abilities and what the olfaction system can do to physiology, behavior, or emotions, it's actually, we have lots of evidence on this. One thing I always like to mention, although it's an old study, I do still love that study where first the researcher looked at what dogs do when they are tracking a scent. And if you have a dog, you probably saw how they would go back and forth around a scent. So this is, sorry, I'm trying to move my mouse. Here in yellow, you could see the scent here of any animal or anything really. And in red here, you see the movement of the dog. So the way the dog would track the scent is to compare the information between the 2 nostrils. And interestingly enough, if you look at whether humans are able to do this, and to test this, you actually need to blind and make sure people do not hear anything. And they actually got participants to get on their knees and try to track a scent. They did this. And they found that humans can do just as well as dogs, which is fascinating, and also adopt the same behavior. So you could see here in pink the exact same behavior as we could see in dogs. So we would also compare the information between the 2 nostrils and try to find that smell. So with some training, humans can also do this. It's not just dogs, I swear. So yeah, there are fascinating abilities that we do have as humans. Another one that I would like to emphasize is the very close relationship we have between olfaction and emotions. And especially, probably you know about what we call the Proust effect, which is the amazing ability of smell to trigger extremely vivid memories. So of course, any sensory stimulation can trigger memories, but olfaction have this uniqueness in terms of triggering really extremely emotional, vivid, detailed, sudden memories that would come back. And it's called the Proust effect because, as you probably know, Proust wrote about the Madeleine that he eats, and suddenly he's back in his grandmother's place with all the memories that come back to him. So odors have always been considered as really potential trigger of emotional reactions. And there are different quotes that I like to also emphasize. If you do consider cognition and emotions as 2 different phenomena, you could also agree that smell is maybe to emotion what sight or hearing are to cognition. So that it's extremely emotional sense. In terms of function of olfaction, you may wonder, but why do we actually still need this sense as humans? We can rely on vision, we can rely on audition, but there are really important functions that olfaction still does for us. And there are 3 of them that I'm going to detail in different subparts, but you can see already if you look at the red ones that emotions are really a very interesting part of the olfaction system. So first, obviously olfaction is extremely important to food intake. So if you probably all did this experiment when you're in the street, you're kind of hungry and you smell this amazing like baking bread smell, croissant, chicken, you choose whatever you're craving right now at 6 p.m., probably something savory. So cheese, whatever you like. So really the olfaction is what allows us to detect a food, for instance, in our environment and to identify it. So it's extremely important for that. It's also important to detect any violation in our expectancies. For instance, if you see ice cream and you think, it's going to be really sweet, and you smell it and it smells like salmon, you may think, okay, it's not ice cream, it's salmon mousse. Or if you look at a fruit, you're not sure it's still good, you may smell it to make sure it's still edible. So olfaction really helps us figure it out if we can still eat this, especially if we have expectancies. It's also important for appetite regulation, so olfactory thresholds are going to change according to hormonal levels, for instance, according to hunger. So obviously the thresholds are going to be really low if you're hungry, so you can detect really small concentration of food, and then they're going to increase while you're eating the same food. And last but not least, it's also important for babies, newborns. Babies are heavily relying on their sense of smell to detect breast and be able to be breastfed, for instance. So ingestion is essential. Obviously, we need to eat to still be alive. It's also important to avoid any danger in our environment. This is a bit different from ingestion, but also extremely relevant. For instance, it could be anything that elicits fear. So this could be the smell of fire or the smell of a gas leak in your apartment. If you cannot smell, this could actually extremely be really dangerous because you're going to smell fire when it's way further apart than when you see it. Same with gas leak. There is no visual cue you can use here. If you do not smell it, you're going to be in big trouble. And it could also be disgust related, so anything that would more relate to microbes, so anything which is vomiting, anything that is decaying, like an animal dying on your garden. Some body odors could also elicit disgust, so anything that would relate to disgust. And finally, and this is a field we understand a bit more now over the past 20 years in humans, but still not fully, social communication is another aspect of olfaction that is also increasingly studied. And it could be important for reproductive purposes, so to be able to detect someone who has ideally an immune system which is compatible with yours and will probably maximize chances of survival for your offsprings. And also for emotional contagion, and I'm going to talk about this a bit later in the talk, we would be able to detect emotional state of someone based on their body odors. And that's probably what we're missing when we are using Zoom, for instance. We are not able to use those information. If you're wondering why this may be that olfaction and emotion are so related, there are different hypotheses. Biologically, it's important to know that the areas in the brain that process olfaction and emotion are very similar. And you can just focus here on the 2 first columns. You don't need to look at the 2 last one. There is, for instance, here, I'm sorry, I'm slow. The amygdala, I don't know if you can see it without my mouse. Here we go. Amygdala is here. You could also see interior cingulate cortex here. Orbital frontal cortex is here, around here, and insula are just areas that we know for emotional processing as well as for olfactory processing. So there is really an overlap in terms of brain areas. If you're wondering how on earth we can study this scientifically, I just wanted to open a parenthesis of, we are lucky enough here in Geneva to have 2 tools to do this. So we have what we call an olfactometer, which is basically a device that allows us to deliver smell in the scanner. So while brain activity is recorded, we can present different smell to a participant. So each jar here actually contains a smell, and the smell is going to deliver through the tubes that you can see up to the participant's nose, and we just use a very small air influx to bring the smells to the participant's nose, and then they can lie down in the scanner, receive smells, and we measure brain activity. And FYI, we also have the same, like a little brother for the olfactometer, which is called a gustometer, which allows us to do the same with actual food. So we can also present liquids in the scanner. So this could be water, syrup, milkshake, alcohol, you name it. And same thing, we'll have the liquids and they would arrive directly in the participant's mouth. So we can also ask participants to actually swallow food while they're lying down in the scanner. And we can obviously combine the two. So present, for instance, the smell of chocolate and then give them a milkshake of chocolate and study brain reactions. In the science literature, there is also traction, and there have been several now special issues on emotion and olfaction, or on food intake and emotion. So if you're interested in this topic, I strongly invite you to look at those. But for this talk, I would like to talk more now on social communication and odor perception. And this is one field which is a major complaint of patients losing their sense of smell, so that's also why I want to emphasize it. And probably you did notice during COVID that we stopped doing all those greetings where we touch each other's nose, either directly like in those pictures, but here in Geneva we obviously cheek-based, which obviously give us a lot of smells. Even handshaking could be associated with a smell. If you look at this study, they actually investigated what people do after shaking each other's hand. And
this is an experiment from the control study.
I actually don't need the sound.
Concurrent recording of need.
This is actually a participant who is lying down here. You could see here on the bottom the way she's breathing in. And you can see an experimenter is coming in, they're shaking hands. They are talking for a bit, so her respiration is kind of flat if you look at the line in the bottom. So they chit-chat, blah, blah, blah. And then you're going to see that when the experimenter is not gone yet, she's going to smell her hand. Just strongly advise you to pay attention. See, she's gone. Here she goes. She's starting sampling her hand. And if you observe people on the street shaking hands, this is a very common behavior. I strongly invite you to pay attention next time someone shakes hands in front of you. People do this almost systematically. And not just that, they generally do sniff each other of oneself a lot. There have been a recent study on this in 2020, like asking, I think it was 400 people, how often do they smell different parts of their bodies, different part of their partner, strangers, kids. And so the redder it is, the more often we do smell that part. And you could see, for instance, for children, women will smell them even more than men, but men still smell them very often. And that's true for our own body odors too. So we tend to check our own body odor a lot. And there has been, I think, now new evidence of why this may be. And already historically, there is this old philosopher who say that every man has confidence in those of the same smell. So this could be one reason why we keep smelling each other. We are trying to find out who is similar to us besides other information. So it's really poetic, but it's also true scientifically. If you think in terms of evolution and threat and security, obviously strangers have the highest potential for threat. I'm not saying all strangers are dangerous, right? I'm just saying in terms of potential of danger, it's quite high. You have neither genetic nor learned connection with them. With relatives, you have a genetic connection. With yourself, obviously, it's the highest you can imagine. And you also have a learned connection with, for instance, friends. So there is all kinds of intermediate solutions here in terms of threats and security. And it's not even mentioning that diseases, besides wars, have been one of the biggest cause of natural death, if you think in terms of history. So to the point of people arguing that different smells could actually be able to be indicators of diseases. So Hippocrates, for instance, said that we could be able to diagnose disease in smelling people. Think of this, think of our body odors as like the olfactory counterpart of fingerprints. It's kind of the same thing, except that the body odors have kind of stable information, such as gender, but also more transient information, such as are you sick and what did you just eat yesterday? And if you're curious to know, in terms of comparison, for instance, thyroid fever would smell like baked bread. So someone who has that disease would smell like baked bread. Scrofula, which is a type of tuberculosis, would smell more like a beer that has been stale. And yellow fever would smell more like a butcher shop. So think of it next time you go grocery shopping, it might just be the people around you, not just the food. And fascinatingly enough, we are able to detect if someone is sick This has been studied in a laboratory context when volunteers got injected with something that would infect them, and the body odors were taken and presented to receivers who had to rate how intense this body odor was, pleasant and healthy. And what you can see in gray is the ratings for the smell of like a body odor which has been infected with disease. And you can see that participants who rate those smells as more intense, so stronger, less pleasant, and also less healthy than if you've just been injected with a saline solution, so something that doesn't do anything. So humans are able to detect early cues of sickness from body odors alone. And in that respect, a human olfaction may just be a signaling route for a behavioral immune system. So it's important to also think that it gives us important information on whether someone is sick or not. Other new information on why we may smell each other like constantly might be finding out if we can be friends. This is a really recent paper on whether there is chemistry in social chemistry. And the take-home message, and I'm not going to go through details, but the take-home message is the researcher used, what you can see here on the left is an electrical nose, and also what they did is really test how similar what we call click friends, which you can see here in the red dashed line. Click friends are just friends you meet and you immediately feel close to, you feel a strong bond with them. Those are click friends. And you can see that for click friends here in red, their body odors, so if I'm clicking friends with someone here, we may have very similar body odors. If you check, the electrical nose is going to consider those two body odors as very similar. And if you ask humans now to rate those smells, they're also going to consider them as really similar. So differences between those body odors tend to be really small, which could be an argument for saying that we may also choose our friends based on how similar we are in terms of body odors. Interestingly, too, is what happens when we are looking at disorders. So, for instance, autism spectrum disorder, which you probably know. And there has been few studies on how they might process body odors. And interestingly, too, they may show different responses to those body odors, especially if you, for instance, present them with the body of odor of smell of fear. So you take the sweat of skydivers and you present them to adults with autism spectrum disorder or normally typically developed adults, you would find really different responses. So typically we find increased arousal and decreased trust for the smells in typically developed adults. We found the opposite in adults with autism spectrum disorders. So it's not that they cannot detect them, they can detect them well, but they may react emotionally differently than a typically developed person would. So body odor do convey social information, although we may not necessarily realize we keep smelling each other, right? It's not something we're typically conscious of, or even if a smell is present in this room. But we could identify kinship, age, gender, hormonal levels, sickness, and emotional state from olfactory cues alone. I want to emphasize, I'm not saying pheromones exist in humans. This is not what I want you to get from this. Pheromones are defined as chemicals that will transmit information to other individuals and will evoke physiological or behavioral response that cannot be modulated in any way. To this day, there is no evidence of such chemicals in humans. It does not mean body odors do not influence us. It just means we may not have pheromones in humans. So please do not spend money on all those perfumes that always promise to make you incredibly attractive that might not just be working. Finally, I would like to say a few words on what happened when one loses his or her sense of smell. And particularly, I would like to mention this study that was done before COVID, but the complaints are very similar in COVID, so I didn't think there was a need to add another one. I like this one because it's a very large one. And to give you rough numbers, about 1% of the general population cannot smell, so we call this anosmia, they cannot smell anything. And about 5 to 8% of the population have reduced olfactory function, so it's fairly common to have troubles with olfaction. And what people report is typically problem with hazard avoidance, so not surprisingly, we find again the complaints related to the function of olfaction. So about 72% of participants were worried about being exposed to dangers, such as fire or gas leak. There were also lots of problems with food. So accidentally eating spoiled food is very common. Think of your fridge. It's been in there for 3-4 days. You're not sure you can still eat it. You tend to smell it before eating it. You cannot do this if you don't have your sense of smell, obviously. People also say they don't really feel any pleasure in cooking anymore. This becomes a big challenge, especially if you have a family to cook for. And about 64% reported a really large decrease in enjoyment of eating or drinking. So this is also affecting their everyday life in a very strong way. Another problem you may not think of is problem with managing body odors. Think of the heat wave. Do you actually need to shower? Are you clean? Can you actually still wear this t-shirt that has been there for a day? Does your kid smell? Do they need to change diapers? Do your house smell? Like, do you need to clean it in some way? All of those questions you cannot really answer to without sense of smell. And people do feel extremely insecure about this and report really awkward situations where someone would come and say their babies need to be changed and realize it's been hours, or the house smell like something died in there, and not have no clue at all. So I do know a patient who said they would take 10 showers in the summer to make sure they do not smell. This goes to those extremes. And another one which is not really well known and not well documented, I find, is social isolation. So most people do not understand how it feels to live without a sense of smell. If you see a blind person on the street, you're going to help him or her crossing the street probably. If you're someone who is deaf, you're probably going to make sure you're articulating well and facing them. But if someone tells you they cannot smell, it's kind of hard to imagine what it must be like. And people report feeling extremely isolated, not understood, have no feeling of empathy, and being really in awkward situation in restaurants and wine tasting where they feel absolutely not understood at all, to the point of avoiding the situation altogether. So people who would typically share this information, I have no sense of smell, would stop doing this with strangers because they did not feel understood at all. And finally, and this is also important for clinical purposes, about 40%, even more, of the individual with reduced olfaction or no olfaction reported decreased well-being, decreased mood, and decreased satisfaction with life. So this could be not smelling your loved ones, your kids. It could be not smelling, I don't know, mushrooms when you walk in the forest right now, not smelling the smells of the spring when the flowers start showing up. And I think it was about like 66% of people said they felt more anxious and vulnerable than before having lost their sense of smell. So it's a lot. Not mentioning a lot of people could also develop depression. Hopefully, my hope, and I am glad we had Julie's talk right before, the COVID pandemic may have changed this belief. There is more, I think, media coverage now on why olfaction might be important and why people who lose their sense of smell in the long term are suffering. But I do really strongly encourage to show more empathy to those patients, for sure. And to conclude, I do hope now you believe that poor olfactory abilities in humans is a myth, complete myth. Olfaction and emotion do go hand in hand. Pay attention next time you have a smell that brings you a lot of emotion. And we are only, I think, discovering now how chemosensory perception may mediate social communication in humans. We have more data in animals, but I think we're still going to discover a lot in the next 20, 30 years. Thank you very much for your attention.
It really was hard to grab our attention at this moment in the afternoon, but Geraldine really succeeded. Thank you for opening our noses to the richness of the olfactory world. We definitely go back home different than we arrived. And we have covered a lot of ground, but we are reserving a treat for the end, quand même. This treat now comes from the Netherlands / the UK. We have Professor Karian van Rekum, who is a professor of psychology and neuroscience at the University of Reading in the UK. So again, this interdisciplinarity that we like to foster. She trained in the Netherlands, the University of Amsterdam. She also trained here in Switzerland, and then in the US at Wisconsin-Madison with Richard Davison, that I'm sure you know, prior to joining Reading in 2007. Her broad range of interests include examining psychological and brain mechanisms of adaptive responding to emotion and their consequences for well-being, how emotional disposition is related to biased processing of information, how individuals differ in their ability to control their emotions and how this changes with advancing age. In terms of methods, she uses measures of brain imagery, so MRI, EEG, and peripheral psychophysiology, facial EMG, skin conductance, heart rate, and in addition, behavioral measures and self-report. Basically, methodologically speaking, if it exists, Karin is an expert on it. And she's also impressive in her commitment to emotion research proper. She has authored over 70 papers, most of which are all on emotion. She was co-editor of one of the oldest journals devoted to emotion. I'm talking about cognition and emotion. And she's one of the co-founders of the annual Reading Emotion Symposium. And all of this, in a way, it's an honor for us because she did her PhD here in this house with Professor Klaus Scherer, who is the founder of this institution, of the Swiss Center for Affective Sciences. So honestly, I can say in everyone's name that it is for us a true pleasure to welcome you back home. Voilà, so she's also very engaged in education of the new generations of affective neuroscientists, something that is close to our heart as well, and in translating scientific findings into everyday insight. And this is what we're doing today. I think that what she's going to be talking about is this skill that is crucial for our well-being on our daily basis and how we can keep it fleshed and alive. This is massively important. Welcome home, Karin, and the floor is yours.
Thank you so much for inviting me and actually to give me the stage here to close what I think has been a fantastic day and a fantastic series of talks. And I'm really, truly honored to have been invited to be part of this. So thanks for that. Yes, did my PhD here in Geneva, or as I sometimes call it, my academic adolescence was here in Switzerland. But really, what Klaus Scherer has taught me still forms quite an important part of my work until today. As this picture illustrates, individuals differ widely in their emotional responses sometimes to the same situation. Whereas the kids generally seem to be rather scared by this thrill ride and by the prospect of being hurtled down this hill at relatively high speed, the adult here seems to be able to see some fun into this whole situation. So why exactly, how come, are the emotional responses of those individuals quite so different? They all find themselves in the same situation after all. So let's break this down a little bit. I experienced a situation, I arrive at Heathrow Airport and all the flights, or a number of flights, are cancelled, including the one that I was about to take. An emotional response ensues. But is it the situation itself that gives rise to a change in facial expression, a change in bodily experience? Heart rate changes, sweating starts to change, muscles start to tense, a change in motivation. I feel like I want to scream. And as well as a change in subjective feeling, or is it something else? And what I've learned here from Klaus Scherer and also at my time in Amsterdam, that process is not per se driven by the situation, but by the meaning that I adhere to that situation. It's not the situation itself, but if I interpret that situation as obstructing me to get to a meeting or a talk in time, that is when I start to experience some negative emotion, such as, the dread here. So the good news is, if it is the meaning that I adhere to a situation, but not the response, or not the situation itself, I can modify the meaning, right? So I can modify my emotional response. And that's a process we call reappraisal. You reappraise the situation in order to change the emotional response that you have. It's not the end of the world. There are more flights to Geneva. I can catch a later one. If I don't show up for dinner, nobody will be upset with me. And therefore, the negative dread starts to dissipate. Okay? So it's this process of appraisal during my PhD, but of reappraisal more pointedly that I've been studying with my colleagues over the last number of years. This work has been heavily inspired by the excellent experimental work and theoretical thinking on emotion regulation of James Gross at the University of Stanford. He's inspired myself and many others actually to study the different emotional regulatory processes. The one that we seem to be focusing all on is that of reappraisal. So contributions from my lab include indeed some of the brain mechanisms that support reappraisal, that support adaptive emotional responding, where we really see that, ooh, my pointer is stuck now, there, where we really see that successful reappraisal really engages some of those brain cortical areas that are also involved in some of our more cognitively complex thinking, our ability to inhibit irrelevant information, task switching, multitasking, those sorts of things. Those regions are also crucially involved in the process that we call reappraisal. Some of our psychophysiological responses, as Christine also so nicely said, also show downstream effects of reappraisal onto the body and also at the same time really are objective measures of showing us that this process of reappraisal is quite effortful, it's quite cognitively, mentally taxing. And here I want to highlight really findings of a very recent study. I won't walk you through what may be looking to some of you as a relatively complex set of data here. I'll try to simplify the story here. This is a study on pain performed by Norma Rosenek, who recently defended her PhD at our university. Collaborating with my colleagues with expertise in pain has taught me actually quite a bit. I myself had I had underestimated the extent to which our pain sensation and our responses to pain stimulation are so psychological, so mental, so closely intertwined with our emotion. I always took pain as being separate from emotion. And my colleagues have taught me that I was wrong in thinking that. It is actually very closely intertwined. So Norma Rosenek, for her PhD, was really interested in trying to tease apart this whole notion, or trying to study this whole notion of the comorbidities, ultimately the co-influences that emotion and pain have on one another. So this is actually a lab-based study that she did that I think may have some consequences or some take-home messages for you as well. Make a long story short, in the lab she delivered pain to people on their arm whilst also manipulating the emotional context. One group of people she invited to come to the lab and get trained on reappraising that pain stimulus in order to change the pain intensity that they may be experiencing. Another group of people she really trained on reappraising the emotional context in which the pain was being presented. A controlled training that a third group of people received was just on something quite mundane, such as toothbrushing, it was in her case. So what she showed in terms of self-report, people come in at a baseline, they get pain presented, they give some ratings on pain and ratings on distress. And also, we measure the extent to which there is a region on their arm around the area where we deliver the pain and calculate how big that is. So baseline, that's all the same. Then we repeat this. They receive the training and they come in for a second session of measurements, and a week later, they come in again. Both the groups that have received reappraisal training, so they were taught to change the meaning or change the interpretation of either the painful stimulus itself or the emotional context that they experienced, we noticed that both of those groups not rated their pain intensity as being lower, rated their emotional distress as being lower relative to the control group. But what I think is the coolest finding of this study is the downstream effect on the actual pain sensation around the pain stimulus that has reduced in both the reappraisal, pain-focused reappraisal and emotion-focused reappraisal group relative to the control group. So if you want to be flexible and you try to reduce that painful experience that you have and you're failing at that, try to regulate the emotional experience that you have at the same time. And that hopefully is helpful. Switching gears a little bit to aging. As I just highlighted, that successful reappraisal, in order to engage, in order to succeed with that, really does rely a bit on this recruitment of those cortical areas that support mental agility, higher cognitive function or cognitive control, the ability to switch, the ability to multitask, the ability to inhibit irrelevant information. So if reappraisal is dependent upon that, what happens as we age? As we know, when we age, our memory goes down a little bit, gets a bit worse. Our speed of processing slows down as well, and so does our mental agility. Our ability to inhibit irrelevant information and to multitask also decreases as a function of aging. That process of aging and that loss of cognitive control and memory is also associated with brain matter loss that is particularly going down at a relatively faster speed in those areas that support cognitive control compared to some other areas in the brain. So you would say, if reappraisal is dependent upon these cognitive control, on the mental agility, on cognitive flexibility, as well as on those brain areas, then we, are not as emotionally flexible anymore as we get older. Well, as we know, you don't need me and you don't need any psychological experimental data to let you know that as we get older, that is not the case. We know that from people who are perhaps a bit older than us, that their emotional function is purely, quite well intact. And in fact, we can learn a lot from older adults as well. So what is going on there? Well, some recent work from us has suggested that also when we look into the brains of adults across a large age range, that we see is that with increasing age, and then when their structure, the brain structure really changes, where they lose some of that grey matter, what we see is that there is a compensation. They start to engage a different brain network. Has no consequences on their emotional function in the end. They get there. They experience the same emotion. They live the same emotion. They're equally flexible with their emotions. They just use, in their brain, a different network, a different process in order to achieve that. Psychologically, it's also that has been found that older adults tend to use different and less cognitively or mentally taxing emotion regulatory strategies, and acceptance is one that apparently, as we get older, and in actual fact I can speak to that, that is something that we learn a bit more of. So we maintain our emotional function by regulating our emotions differently and staying in that respect cognitively and emotionally flexible. What does happen, and that's one of our recent data sets, first authored by Will Lloyd, a postdoc who's now in Manchester. What does happen, however, is if that cognitive decline really starts to decline quite steeply, and if our executive functions particularly have declined over a number of years, that at some stage does start to affect our ability to regulate our emotion. We see it also quite markedly for those of you who know an individual with neurodegenerative disease, such as Alzheimer's disease or other forms of dementia. You may know that they may avoid stressful situations, but once they do find themselves into it, they find it harder to regulate. So there is certainly, at some stage, a tipping point in our ability to be flexible with our emotions and to regulate them as the context calls for it, or as you yourself desire. That tipping point in emotional flexibility, however, if you ask me, is much later, is delayed relative to the tipping point of a change in our cognitive function and our brain structure accordingly. So that's the good news. And I think actually that we can learn quite a bit from older adults as well in terms of flexibility, particularly in the emotional domain. The other thing I want to quickly highlight, and I hope time permits me to do that still, is that, this is all great, it's all grand, it's emotion regulation in order to down-regulate particularly negative emotion, starting to feel better, promoting happiness. That is all nice and good, and it's great that some people can do that, but it's not really great if that doesn't happen for you. So individuals differ in the extent to which they can be emotionally flexible. And even in our own, in our own lives, there's some contexts where we find it easier to do this, to perform this emotion regulation than others. And, we don't often have somebody tell us, like, okay, now decrease, and you know how you can do that. There's nobody really on our side, in most cases, to help us train through that. So individuals with depression, as I already highlighted with some of our brain imaging data, have trouble doing so. And in fact, they try to recruit those cortical areas, they try to reappraise, and it's actually for them counterproductive. That's what those brain data also showed. So there's different also situations and challenges that we may be presented with that make us harder for us to do. There's one it's depression, it's anxiety, neurodegenerative disease, a change in cognitive function. COVID may even be another one as well, hopefully temporarily. So one that I really want to focus on is intolerance of uncertainty. Jane Morris has been spearheading in my group that whole work on intolerance of uncertainty, and has her own lab at the University of Southampton, where she started earlier this year. She's performed this work using threat conditioning and threat extinction work, and I'm going to threat conditioning and threat extinction tasks, and I'm going to illustrate what those tasks are by way of example. This is a roundabout very close to my home. Drive on it every time I'm in my car, which is not every day, but frequently enough. Think nothing of it, of that roundabout. Until about a year ago, I found myself on that roundabout. Car came from the left, decided not to stop, hit me in the side of my car span, and the car was a total loss. But I was totally fine. So, okay, well, that all got sorted. So the next time that I approached that roundabout in my now new car, I did start to notice, and I'm not a particularly anxious person, I did start to notice that I started to tense up, I started to feel my heart rate, and I started to pay attention very much to the left of those cars that are approaching from there, and I couldn't let go, right? So what I'd done there is I learned fear. So threat was conditioned, fear was conditioned, I'd acquired fear. So this context became threatening to me. So that's indeed what Jane Morris did. She had a lab-based task of this real-life situation, and all the individuals that came to those lab-based tasks where she measured psychophysiology in addition to self-report, she also asked people to complete various questionnaires to measure their trait anxiety, but also to measure their intolerance of uncertainty. She used those skills that include questions such as, I must get away from all uncertain situations. Uncertainty makes me uneasy, anxious, or stressed. So as those questions illustrate, intolerance of uncertainty is highly comorbid, or it overlaps a lot with anxiety and with anxiety disorders, and that's definitely the case. And some of my anxiety experts colleagues have already said, like, is intolerance of uncertainty not just a different word for anxiety? No, because not all individuals with anxiety and even anxiety disorders experience intolerance of uncertainty. So we are really trying to understand some of those mechanisms that are transdiagnostic, that are not just common in anxious people, but also people experiencing some symptomatology of autism, for instance, or ADHD. So we really want to understand some of the mechanisms, and that's why we really focus on intolerance of uncertainty in addition to trait anxiety. So individuals who are highly intolerant of uncertainty respond the same way. I mean, it's adaptive that I learned to fear this context, right? So everybody really pretty much responds the same way. It's what happens next that is really, really different. So in my new car, I now come for the second time at this roundabout. My anxiety really starts to reduce. I do it the third time and I do it the fourth time, and by that stage, by the fifth time, this roundabout is like any other old roundabout for me. Individuals who are highly intolerant of uncertainty, so what I've done, sorry, I need to make a step back, is I've extinguished my fear, I've extinguished my threat. That is an active process. It's often being thought of as a passive process. It is not. It requires updating my memory of the threat, that something is now safe. So, again, you need to be a bit flexible. It's a very implicit process. You're not very aware of it, but you need to update that threat. memory for that it is safe. Individuals who are highly intolerant of uncertainty have trouble doing that. It's not so much reflected in the self-report, but it is reflected in the bodily measures that we take, specifically skin conductance, a measure of bodily arousal. So highly intolerant people, high intolerance of uncertainty people have delayed threat extinction. It takes them much longer to update that memory. So they're less flexible to update that memory of threat. They respond, especially in terms of bodily arousal. They respond also quite strongly with bodily arousal in situations like uncertainty as well. And they generalize, not just that roundabout, but any other old roundabout starts to become a threat. Although, quite frankly, you don't need to be too anxious or too intolerant of uncertainty to be a bit intimidated, what the Brits call the magic roundabout, where you have to get onto a roundabout, fire a roundabout, and try to find your way off the roundabout with a roundabout. So that's normal if you're being intimidated by that. So individuals high intolerant of uncertainty have difficulty updating that memory. They take much longer to do it, respond more strongly with their bodily response. And it is also quite specifically to situations with negative variance. So a surprise party, individuals high in intolerance of uncertainty have no problem with the uncertainty that is related to that, of whether that is going to happen or not, etc., etc. Not a problem there. It's really quite specific to negative situations. They respond like any person in this experience, in those types of experiments, when we instruct them, when we let them know, like, look, listen, there is no uncertainty here. There is no threat that's going to come. That car is not going to smash into you. Once they're being instructed, so they get some help from, like, external help, then that starts to, their uncertainty is gone and they respond like anybody else. So they can respond like everybody else. It's just that they can't self-generate. So it's the difficulty with flexibility and self-generating that safety memory, that level of learning. So how am I doing for time? 5-ish minutes? Okay. So can we train ourselves then to be more emotional and flexible? I've already gone through reappraisal. It is something that you can do at home, but that reappraisal is also very much a hallmark of cognitive behavioral therapy, which is used to treat, for instance, anxiety and depression, among other mental health issues. So yes, by training yourself, if you can manage to generate that, by all means, re-engage with that. What some of the findings have shown in the literature as well is that mindfulness can be helpful to train your emotional flexibility. That behavioral activation, specifically for those individuals who may experience depression, try to avoid situations that they don't want to expose themselves in or want to find themselves in, but really trying to overcome that hesitation. It can really help you to overcome any feelings of depression and improve your emotional flexibility. Exercise, I've seen some work on exercise today as well here at the poster session, has been shown to improve not only your cognitive function, but also sometimes your well-being, so that can certainly help you. Diet, due to my colleague Claire Williams, who's been studying the effects of blueberries, of which I was always being quite skeptical in the extent to which that may have some benefits. She's shown that that can improve complex cognitive function, and also we're now collaborating on how it may improve depressive symptomatology in some positive affect. The effects are really small, but as a result, I now start my day with a handful of blueberries in my yogurt. Biofeedback, particularly heart rate variability. You can also, if that's something that works for you, you can try to engage with that in order to really get nice variable heart rate, which is good for emotional flexibility as well. And cognitive training. I want to say one final word on that. All of these, I'll get back to that. Let me get back to cognitive training. You've probably heard from Nintendo, brain training, and it is controversial. The extent to which, you can use those apps or use any cognitive training regime, the extent to which that actually transfers and has benefits for cognitive function in your daily life, and by extension also for your well-being and emotional function in daily life, has been relatively controversial. You get good at the task, which is fantastic, but are you getting better at that, at other aspects? So there's been some doubt about that. Naz Direction is a colleague that recently joined us at the University of Reading. She has been using working memory training regimes where people for like 2 or 3 weeks, for half an hour a day, or something akin, or a few times a week, do indeed some working memory tasks. She has shown that individuals with anxiety, it can help them reduce their anxiety, particularly women with breast cancer who've just been diagnosed with their breast cancer. It helps them cope with the anxiety and depression that they feel as a function of the diagnosis and as a function of their disease. It surprised me, and she always admits herself, it surprised her as well, but it does help. Bilingualism, one of my colleagues is studying the effect of bilingualism. I thought to highlight that in a country in Switzerland where people at least speak two languages fluently, in my experience, confers benefits for cognitive control, executive function. So by the end, that may also help in terms of helping cognitive flexibility as well as emotional flexibility. So when to perform the training, just engage with anything that you may like the look of. Certainly in moments, does it help you? I don't think anybody in the room, given that you've been motivated enough to educate yourself about emotion and really sit here, I think you're already flexible enough as it is. So keep going, quite frankly. But if you experience challenges in your life, if you perhaps get a bit older and you start to notice that your brain is just not working so well with all that information that comes to you, pick one, I would say. Because is there one that works better than the other? Pick 1 or 2 or 3 that you like the best look of. At the same time, we really do, as a group of psychologists, really work out better what is going on with all those different interventions, where it is helpful and where it can hinder us, and to whom this may confer most of the benefits. Hope you enjoyed it, and thank you for your attention.
Argue around the artists asking aspects to avoid, challenging change, choice and voice. In a cognitive context, daily decisions develop the dream, encounter, environment, events, experience. Faced feelings, few findings. Ideal imbalance, important intolerance involved, needed for other problems. To reach resilience, seeking sense, shaping ships, to think and sing in those times, to undermine, will be young, 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 young. Encore une fois. To argue, to ask, to avoid, to be, to challenge, to change, to choose, to choose, to develop, to dream, to embody, to encounter, to experience, to face, to feel, to find, to imbalance, to import, to involve, to reach, to seek, to ship, to think and sing, to time, to undermine, to voice, to will.
Olfaction, cholinergic signaling, populism, long COVID consequences, empowering communication, gender-specific brain characteristics, mental health, anosognosia, emotional flexibility, the decomposed words and the recognizable ones. What an incredible tour de force in the study of emotion we have had today. I would like to thank this incredible presentation conducted by stunning researchers and stunning communicators today. I would like to thank our artists for pushing us to feel in different ways. I would like to thank the organizers of this incredible series. So, of course, Viviana and her team, both BrainCircle Italia and Lugano. And I would like to thank the local organizers as well, who have done a stellar job in bringing us together, Marcello Mortillaro, Carol Baron, and Marion Gumi. I would like to invite them all to come here if they feel so inclined, so that we can close this together physically, since we know that smelling one another is important. So if you don't mind, maybe join me for the last words, so that people can see you who were involved. I think Carol and Marion are not with us, but...
You still trap me into coming. So, let's say, okay, so you managed to make me say something. Sorry.
I will finish, but I'd like you to see a face of one of the people who are responsible for us being here.
How do I turn it on? C'est bon? Voilà. Carry on.
Thank you. Thank you very much. Viviana, would you like to join us too? You are the organizer of this just as much as we are. Absolutely, absolutely. I would like you to be able to see the faces of those who made this possible. Absolutely.
I don't know. I just want to thank you all for being here. I think it was an incredible journey. I want to thank Marion and Carol, actually. Please come forward, please. You shouldn't be shy of that. And so I don't want to talk to be the last speaker. It's been women. It should be not me who finishes. So I give you the microphone and then to Christina.
Well, thank you. I have not much to add, but one thing, yes, I want next time to speak also about auditory, because with the music made me think how much audition, if that's the right word, increases our emotion or changes our emotion, how important it is in sickness and health and happiness. We know that, listen to music can really do a lot, for example, for Parkinson and Alzheimer. So we didn't think about this, but this is a good thing for next meeting to have that included also. And I thank everybody for being here, and you and David and all of you for hosting us and making it such a special event. It's a great conclusion for the Emotion conferences. I'm very happy to be in Geneva with you.
Thank you.
Thank you so much. Well, in this great company, I will then close the event. I told you at the beginning this was a celebration of science, of emotion, of women, and with the proper emotions that accompany a celebration we leave. So gratitude, joy, unity, inspiration. Thank you all very, very much.